Hello, my name is Solomon Habt Mariam. I'm a leader of Herbal Analysis Services UK and Pharmacognosy Research Laboratories at the University of Greenwich. To my research collaborators and my website visitors and followers, I have a little presentation to make today on repurposing chloroquine and hydroxychloroquine for COVID-19. By now, you all know that this subject has become a center of discussion in recent weeks. I want to briefly summarize what we know, but most importantly, what we don't know and should know about this approach. As always, nature not only serves us as a valuable source of new drugs, but also inspires us to make more potent, less toxic, and easily accessible natural product derivatives. The discovery of chloroquine in 1934 by German scientists is just a good example. It was a derivative of quinine which was extracted from cinchona tree barks, and the crude mixture was used as anti-malarial drug for at least 2 centuries before chloroquine. Hydroxychloroquine, as the name implies, is a little more polar derivative of chloroquine, which was introduced a decade later and shown to have less toxic side effects. Despite the drug resistance problem, these two compounds have been used for decades as safe and cost-effective medication, primarily as prophylaxis for treating malaria. Through serendipity and trials, they also found use in autoimmune diseases such as rheumatoid arthritis and systemic lupus. The focus now is on the claimed use of chloroquine and hydroxychloroquine for the treatment of COVID-19. As you can imagine, the desperate need for therapy of this new disease now appears to push governments and their political leaders to rush into using them for COVID-19. To put this into perspective, there are very limited scientific evidence even in vitro. There are at least a couple of publications showing the effect of these drugs against the virus SARS-CoV-2 in cellular models. They seem to act at micromolar concentration range with some degree of selectivity to the virus. The clinical evidence is patchy and mostly came from China, where some positive outcomes from case studies and preliminary clinical trials were reported. We also have one French trial, but they should all be taken as preliminary data with limitations in size, methodology, and follow-up studies. The mechanisms of action of these drugs is also largely presented as speculative arguments. The virus enters host cells through two mechanisms. First, it uses its spike proteins to bind with host receptors such as angiotensin-converting enzyme 2, which we may be targeting by drugs such as chloroquine and hydroxychloroquine. Secondly, it enters by endocytosis, where an endosome carrying the virus releases it inside the cell by the action of lysosomal enzymes under low pH. Chloroquine and hydroxychloroquine, being basic compounds, are known to raise the intracellular pH and hence assumed to inhibit viral release through this process. The anti-inflammatory properties of these compounds have also implication to the very late stage of COVID-19, which is characterized by cytokine storm leading to pneumonia, respiratory distress, and organ failure. While these mechanisms could be debatable, on the positive side, we know that these compounds have been used safely for decades. We know their side effects, and thus we can manage their use under clinical conditions. With the little introduction we have now on the potential application of chloroquine and hydroxychloroquine for COVID-19, the big question that I want to address here is what no one is talking about these drugs. Our commentary article with my Italian colleagues Marinina and Giovanni was published today in the Journal of Molecules. It is entitled COVID-19, Chloroquine Repurposing and Cardiac Safety Concern. chirality might help. For anyone interested to access this article, they can do so by going to Molecule's website, and it is in volume 25, issue 8, and page 1834. We are calling for revisiting the chemistry of these two compounds to highlight some things that we must do to improve their safety and efficacy. If we look at figure 1 here, both compounds are based on the 4-aminoquinoline skeleton shown in blue with chlorine at position 7 and the diethylaminopentyl group attachment. The difference between the two compounds is the presence of just one hydroxyl group addition in hydroxychloroquine. In these structural drawings, the hydrogens are not shown, and if I add them, as you can see in figure 2, the profile for one carbon is quite different from the rest. This carbon, shown in red, is bonded to 4 different groups: the blue-colored quinoline skeleton, the methyl group, the hydrogen shown in green, and the aminoalkyl chain shown in black. This carbon is a chiral center, and we can draw the methyl group, for example, in figure 3 and 4 here, either above the plane shown by the solid bold lines or below the plane shown by these dotted lines. These are what we call chiral compounds, and the two sister compounds are called enantiomers. We designate them either R and S or plus and minus. For example, S plus chloroquine and R minus chloroquine, or S plus hydroxychloroquine and R minus hydroxychloroquine. The unique feature of enantiomers is that they are mirror image to each other, just like my left and right hands. This means that they are not superimposable. In pharmacology, we tend to see biological effects through actions on receptors, proteins, and enzymes working through the lock and key model. If you see figure 3 in this slide, the enantiomers that fit with the receptor or enzyme active site may act as a drug, while the other that doesn't fit may not be, but instead it could have adverse side effects. Looking into the three-dimensional model of chloroquine will help you to see and appreciate the lock and key model of drug's action. The rotating image on the left shows the planar or flat appearance of the aromatic quinoline skeleton, while the shape of the branching chain could vary. The image on the right shows how the methyl group of the chiral center indicated by the red arrows could either be on the same side or nearly opposite face of the planar ring here. With all this being said on the chirality of chloroquine and hydroxychloroquine, remember, we use them currently for malaria and autoimmune diseases as chiral mixtures. With respect to COVID-19, we don't know whether there is a selectivity difference between these enantiomers for SARS-CoV-2. We just assume that they are equally potent as with the in vitro data for their antimalarial action. But we know that there is some pharmacokinetics difference between the enantiomers in mammalian system. For example, the plasma protein binding potential of chloroquine for the S form is higher than that of the R form. We can also say that the renal excretion is higher for the S than the R form for both compounds. There is also difference in their entry to retinal tissues with implication to their retinopathy side effects. The toxicity in animals has also been shown to be less for the S form than the R form of chloroquine. The cardiotoxicity of these compounds is also well known, and they could prolong QT interval of the ECG, and monitoring their dose is critical, especially in patients with underlying heart problems. This all means that even if the two enantiomers have the same potency as antimalarial and antiviral agents, we should choose the one with less toxic side effects or good pharmacokinetics profile for treating COVID-19. The way forward is therefore to separate the enantiomers, and for this we have numerous options. In addition to the old method of fractional recrystallization, we have seen thousands of chiral stationary phases developed in recent years. In my lecture series, we have seen them grouped into 7 major classes. If you are a good chemist, you can also get these enantiomers through a stereoselective synthesis approach. Once we obtain the enantiomerically pure compounds, we need to test, and test them. This is in vitro in cellular models, in animals, and in humans through well-designed randomized clinical trials. In the meantime, keep cool, be safe, and look after those around you. I thank my good long-time Italian friend and research collaborator, Professor Giovanni Lentini, for his amazing insight in this field. As always, any question is welcome, and you can contact me either through my website links or my email info@herbalanalysis.co.uk. I have also created a dedicated COVID-19 page in my website for an up-to-date information on our activity in this field. Those of you who want to contribute an article on coronavirus area can visit my special issue that I am editing for the Journal of Molecules. Thank you.